introduction. My name is Lauren Hodge. I'm a graduate student working jointly in the labs of Dr. Chris Benner and Dr. Alon Gorin. And today I'll be talking about my project, characterizing the activity and roles of neurological disorder linked to chromatin regulators. So the epigenetic regulation of gene expression has emerged as a process of major importance in cognition and neurological disorders. There's a recent study in Cell where they did exome sequencing in a large number of individuals with autism spectrum disorder, and they identified 102 risk genes, either associated with ASD alone or ASD and other neurodevelopmental disorders. Most of these genes are expressed early in brain development, and most risk genes have roles in either the regulation of gene expression, which are in the boxes in purple, or neuronal communication in the boxes shown in green. And the genes highlighted in the purple box and highlighted in black boxes are chromatin regulators, and these are highly overrepresented in the gene regulators list, illustrating their importance in understanding neurodevelopmental disorders. And so for some brief background on chromatin regulators, chromatin accessibility and the related histone modifications that affect it are functionally intertwined with the activity of regulatory elements and their downstream effects on gene expression. So DNA is organized hierarchically from its double-stranded helix, which wraps around nucleosomes, and then those condense into chromatin and chromosomes. So the nucleosomes themselves are made of histone proteins, and each histone can have various post-translational modifications. So on the right is just an example of histone acetylation, where an acetyl group is added or removed by that HAT HDAC family of enzymes added onto usually the lysine tail of a histone protein. And these modifications can control nucleosome compaction, DNA accessibility, which can obviously alter downstream gene expression, and they themselves can also aid in recruitment of other DNA and chromatin regulators. So they have a key role in general DNA and gene expression regulation. And so in the previous study I mentioned, one chromatin regulator that was linked to autism spectrum disorder with high confidence was SETD5. SETD5 is a histone methyltransferase, meaning it is a writer. It adds a methyl group to the lysine tail of a histone protein, in this case, lysine 36 on histone protein H3, and it's thought to be involved in transcriptional elongation. The figure on the left is from a paper that validated the effects of SETD5 knockdown in mouse models and its effect on autism spectrum disorder-like behavior. However, mice do not always accurately model the human brain development or pathology. So our goal for this project is to use human-based models to gain a better understanding of SETD5's role in neurodevelopmental disorders. And so my collaborators in the Moultrie lab, in particular, Dr. Angels Almanar, generated the first human-induced pluripotent stem cell-derived ASD SETD5-related model. We took cells from an individual with a point mutation in SETD5 on its set domain. The point mutation is it's shown by the green arrow. And then we generated human iPSCs and differentiated the cells into various neuronal cell types. We also generated a rescue clone from these cells where SETD5's function was restored with CRISPR-Cas9. And so for all of the figures in this slide, C is the control, M is mutated, and R is the rescue where the mutation was fixed. And so we see that the mutant neurons have lower levels of certain synaptic proteins, such as SYN and PSC5, shown by the Western blot, and that these neurons also have shorter neurites. We quantified these results as well, shown in D and F, and then we also measured the number of spikes and bursts in the top and bottom in figure G in 3-minute multi electrode arrays recorded in neurons, and observed fewer spikes and bursts in the same interval. So we observed that, like in SETD5 deficient mice from previous studies, these SETD5 mutant neurons also present structural and functional defects. And so, like I said, some of our previous studies and our own preliminary data shows that SETD5 plays a role in transcriptional elongation. And so we hypothesized that the generally reduced transcription elongation caused by SETD5 deficiency, plus the changes in gene expression in SETD5 bound genes, might remodel genome 3D architecture. And so to test this hypothesis, it's really critical to eliminate the secondary effects due to total knockdown. So we basically generated a DTAG system to rapidly degrade allele-specific protein chimeras to have some temporal control. So DTAG system, basically, it tags the protein of interest, and then in the presence of the DTAG13 chemical ligand, the tagged protein is recognized and eliminated by the cell's endogenous ubiquitin degradation pathway. So the schematic of our construct for SETD5 is shown on the right, and we first tested the system by tagging BRD4 and HEC293Ts, and showed efficient BRD4 depletion 4 hours after adding DTAG13. And then after this was validated, we tested this also for SETD5 and evaluated the performance. So we confirmed partial reduction of HA tagged SETD5 by Western blot with either SETD5 or HA antibodies. shown in C and D. And then importantly, one thing we did notice is that HA tag SETD5 does not degrade in the absence of D tag 13. So we have strict temporal control over it. And we can use these constructs to generate homozygous SETD5 D tag tagged human iPSC lines, and then use the A tag 13 to deplete SETD5 in different cell types. Our goal is to deplete in neuroprogenitor cells, neurons, and astrocytes. And so another big hurdle for investigating the role of SETD5 is that there's currently no published ChIP-seq data of endogenous non-tagged SETD5. So tagging, like using our DTAG system, allows great temporal control to knock down and observe immediate effects, but we're also very interested in the binding profile of SETD5 in unperturbed normal cells. Because without the binding profile, it's a lot more difficult to decipher the role in transcription elongation. So I spent a lot of time optimizing our lab's current ChIP-seq protocols, anticipating this challenge. I varied things like chromatin shearing conditions to get the desired length of chromatin fragments, the lysis buffer, antibody conditions, and in the end, I was able to generate binding profiles of endogenous non-tagged SETD5 in HeLa cells. So on the left are genome browser tracks, just showing the ChIP-seq signal for SETD5 at various genome loci, as well as some accompanying histone modifications. So H3K27 acetylation is a common modification showing active enhancers and promoters in open chromatin. And then also H3K36 trimethylation, which, as I mentioned earlier, is the target of SETD5. So here you can clearly see the effects of various rounds of optimization. So on the left is basically just the amount of antibody per IP, the different lysis buffers that we tested, and the different shearing conditions, which, as I said, can basically affect the fragment length of the chromatin. And so testing all of these conditions, we got a very good idea of what would be the best optimal conditions for SETD5. And I identified the conditions in the top row as giving the most robust and clean signal for SETD5. And so most of the SETD5 peaks seem to be located in gene bodies similar to those shown here, which would make sense given our hypothesis about its role in transcription elongation. So to confirm this, I generated metagene plots, and the result is on the right, where the best performing experimental conditions are shown in basically the bold orange, and then the control samples in gray, and the rest are more basically later shown there. So this basically can give us a lot more ideas in terms of figuring out exactly where SETD5 is in the genome, and basically combining this approach. Now I'm going to be combining this optimized ChIP-seq protocol to map SETD5 binding in the SETD5 model discussed previously. So we're going to be mapping at various stages of differentiation and different cell types, so neuroprogenitor cells, neurons, and astrocytes, with or without the point mutation associated with autism spectrum disorder. And I'll also repeat this assay in these cell types with the recent SETD5 knockdown using the DTAG system. And I'll also be combining this with other NGS methods like RNA-seq and ATAC-seq to measure the effects of SETD5 perturbation on downstream gene expression and chromatin accessibility. And then in particular, I'm going to be doing GROW-seq, and this will be really useful because it allows us to assess the direct effect of SETD5 perturbation on alterations in transcription elongation, because GROW-seq measures nascent or newly transcribed RNA. So that's going to be really helpful in basically figuring out exactly where the knockdown of SETD5 and where its binding correlates with changes in transcription elongation. So in conclusion, what we are currently working on in this project is the first comprehensive genomic mapping of endogenous SETD5 in HIPSC-derived brain cells. And the reason that this is really important downstream and kind of what we're hoping to achieve out of this project is that Dr. Angels Almanar in the Motri lab has done considerable research that has identified some key features that differentiate the ASD SETD5 neuroastrocytic environment from the healthy environment. Specifically, where levels of functional SETD5 decrease, the levels of IL-6 and JAK increase significantly, but the mechanistic pathway is still unknown. So by acutely depleting SETD5 in the different cell types and identifying the immediate changes in chromatin structure and the impact on transcription elongation, we can discover unique insights into SETD5's role as an epigenetic regulator and its target genes in various cell types. And hopefully this will allow us to predict consequent short and long-term functional impairments. And I want to thank everyone finally in the Goran and Benner labs, and especially those listed here, and our collaborators, Dr. Motri and Dr. Angels Almanar. And finally, I want to thank the Kavli Institute for funding this and making this project possible. Thank you. Any questions?
Great, thank you. Any questions from the audience? The I might have missed it at the beginning. How did you guys narrow down SETD5 as a target for this? What was the motivation behind it?
So it was basically it was one of the genes I listed initially, basically in the risk factor. Basically, in the study that identified some risk factors, SETD5 is on the top right, where it was one of the genes identified, and it had a very, very strong link. And then also, basically, we had some preliminary data in our lab that made us very interested in SETD5 in particular, because I study a lot of transcriptional regulation, and so SETD5 playing a role and having a clear effect on the neuron's functionality, as well as a basically direct functional role in transcription, is what basically made us focus on it.
And you guys looked at this, and I know they were derived HPSC cells, but iPSC cells, but these were not in intact organoids. These were dissociated cells, or did you actually get brain organoids?
We have just plain plain cell culture types, like neurons and astrocytes, and then we are also working on getting organoid models up and running as well. Yes.
You are great. Okay, excellent.